Imagine for a second that you're trying to manufacture tiny glowing swords out of pure poison.
Right, which sounds totally absurd.
Totally. But then to make it even crazier, you have to do it inside your own stomach, right? Without cutting yourself and without actually getting poisoned.
It's basically a microscopic miracle.
Exactly. And that is essentially what we are looking at today for this deep dive. We're exploring this wild space where the absolute cutting edge of human nanotechnology literally collides with the gritty, I guess, billions of years old survival mechanisms of bacteria.
Yeah, and the sheer elegance of it is just staggering, really. I mean, we spend billions of dollars building these pristine, mathematically perfect silicon wafer clean rooms to manufacture our tech.
Oh, yeah, the sterile suits, the calibrated lasers.
Exactly. We expect absolute predictable control. But nature solves our most complex engineering problems in environments that are chaotic, messy, and, very much alive.
Alive and kind of gross sometimes.
Right. The stakes in nanobiotechnology right now are incredibly high, and it turns out our best engineers might just be single-celled organisms.
It's wild. So for you listening, we are diving deep into the mechanisms of E. coli today. And yes, that is the exact same bacteria that might give you a terrible stomach bug from, like, a bad potato salad.
It's the very same.
But here, they're acting as microscopic factories. They aren't just ingesting highly toxic heavy metals to build these incredibly valuable glowing things called quantum dots, but they've also spontaneously invented a biological trash disposal system to survive the whole process.
Which is the real breakthrough here.
Yeah. By the end of this conversation, we're going to see how this microscopic garbage sorting routine is poised to just completely revolutionize modern medical imaging and targeted therapies.
But to really appreciate the biological alchemy these bacteria are pulling off, we kind of have to ground ourselves in the sheer hostility of the material they're working with.
Oh, for sure.
We are bridging this massive gap between a highly coveted human technology and the incredibly dangerous biology of heavy metals.
Okay, so let's start with the prize. Quantum dots. For anyone who hasn't been, obsessively tracking the optoelectronic space, these things sound like they belong on the hull of a starship or something.
They really do sound like sci-fi.
But they are very real semiconducting fluorescent nanoparticles. And the operational word there being nano. We are talking about structures ranging from, 1 to 10 nanometers in diameter.
Which is an almost incomprehensible scale to the human brain, honestly. I mean, a single strand of human hair is roughly 80 to 100,000 nanometers wide.
Wow. Okay, so we're talking unfathomably tiny.
Exactly. A quantum dot is a cluster of atoms so small that the classical laws of physics literally start to break down, and the weird rules of quantum mechanics just take over.
And that tiny size is the entire point, right? Because it triggers what's called the quantum confinement effect. This is where the physics gets incredibly beautiful, I think.
It really is elegant.
So when you have a bulk semiconductor material, electrons need a certain amount of energy to jump across the band gap. That's the space between the valence band and the conduction band, right? They need that jump to conduct electricity or emit light.
Spot on. And that band gap is a fixed property in a bulk material. But when you shrink a particle down to just a few nanometers, you are physically confining the electrons to a space smaller than their natural wavelength.
You're squishing them.
You are essentially squishing the electron-hole pair, what we call the exciton. The exact size of that microscopic space forces the energy states to widen.
So the size of the room basically dictates the energy of the jump.
Yes, exactly.
And the energy of that electron's jump dictates the exact, precise wavelength of light the dot emits when it glows.
That is the absolute magic of quantum confinement. So say you have a 2-nanometer dot of cadmium sulfide.
Right.
That might glow blue. But if you add just a few more atoms and push it to, say, 6 nanometers, that exact same chemical material will glow red. You are tuning the optical properties, the exact color, entirely through physical size rather than chemical composition.
Which gives us an unprecedented level of precision in medicine and tech. Like if an oncologist wants to track 3 different types of cancer cells simultaneously in a live biopsy, they don't have to rely on traditional organic dyes anymore.
Because traditional dyes are flawed.
Right. They fade. They bleach out under a microscope after a few minutes of continuous observation. But if you tag those cancer cells with 3 different sizes of quantum dots, they light up in distinct, brilliant, unfading colors.
And they will glow brightly for hours or days or even indefinitely.
The utility is unmatched. I mean, whether it is for mapping the the exact margins of a glioblastoma tumor during a delicate neurosurgery, or creating the vivid, hyper-accurate colors on the latest high-end OLED televisions. The demand for these nanoparticles is just astronomical.
It really is. But of course, that brings us to the massive, glaring problem, the peril part of the equation.
Right.
To manufacture these highly coveted quantum dots, you generally need heavy metals. We are talking about lead, tellurium, and specifically for our deep dive today, cadmium.
And cadmium is, well, it's fundamentally incompatible with biological life. The toxicity isn't just a generic poison effect, right? It's a systematic dismantling of cellular machinery.
It is brutal.
Let's unpack the chemistry of that toxicity for a second, because it is wild. It all stems from cadmium's status as a soft Lewis acid, which gives it an aggressive chemical affinity for sulfur, a soft Lewis base.
And biological systems rely entirely on sulfur to maintain structure and function. So the cascade of failures is incredibly rapid.
What's the first thing to go?
One of the first things cadmium does is bind to the sulfur in glutathione. Now, glutathione is the primary cellular antioxidant. It is the chemical bouncer that roams around the cell, neutralizing oxidative threats.
Got it. So it's protecting the cell.
Exactly. But when cadmium locks up the sulfur, it dramatically reduces the total pool of active glutathione, leaving the cell completely defenseless.
And the moment the cell loses that antioxidant shield, it just gets overwhelmed by reactive oxygen species, right? ROS.
Yes, the ROS levels spike.
These are highly unstable molecules like superoxide radicals or hydrogen peroxide that essentially act as microscopic rust. They strip electrons from whatever they touch, whether that's lipids in the cell membranes, structural proteins, you name it. The cadmium induces this massive oxidative stress.
It actively accelerates the decay of the cell. But the damage goes even deeper. It goes all the way down to the genetic code. Cadmium is notorious for inhibiting the mismatch repair system in DNA.
The proofreader enzymes, right?
Precisely. When a cell copies its DNA to divide, the polymerase enzyme naturally makes typos. It's normal. The mismatch repair system follows right behind it, excising the wrong base pairs and fixing those typos.
So cadmium just knocks those proofreaders offline.
It entirely shuts them down. This results in hypermutability. The cell starts replicating with massive genetic errors, which in humans is a direct pathway to highly aggressive cancers.
That is terrifying. And if destroying the DNA proofreaders and basically rusting the cell membrane wasn't enough, cadmium also goes after the cellular engines.
Oh yeah, it targets the enzymes directly.
Because a massive percentage of our enzymes, and bacterial enzymes too, rely on divalent metals like zinc, iron, or magnesium. These metals sit right in the catalytic center of the enzyme, acting like a key in an ignition lock to make the chemical reaction happen.
And this is where cadmium is incredibly sneaky. Because cadmium has a very similar ionic radius and chemical profile to these essential metals, it acts as a molecular imposter.
Sneaky.
It sneaks right into the active site of the enzyme and replaces the zinc or the iron. But because its electron configuration is just slightly different, it refuses to turn the lock. The enzyme is permanently jammed.
The biological engines stall out entirely, and when these proteins can't fold correctly or do their jobs, they just clump together into these massive, useless, toxic tangles called inclusion bodies. The cell essentially fills up with its own broken machinery.
Which forces us to confront a massive paradox in the field of nanobiotechnology.
Right. We are trying to build biomedical imaging tools out of a substance that causes hypermutations, melts proteins, and induces massive oxidative stress.
We desperately need the brilliantly stable light of the quantum dot to save lives. But the chemical process of synthesizing it in a lab yields a harsh, naked, heavy metal crystal that destroys anything living it touches.
Wait, so if these things cause hypermutations and melt cellular engines, why are we trying to put them inside human bodies for bioimaging in the first place? Isn't the cure worse than the disease?
Well, what's fascinating here is that the utility of the dots is so high, we can't just walk away from them. The light is perfect. So scientists were desperate to find a way to make them biocompatible.
We need the light, but we have to neutralize the poison. And because traditional human chemical engineering leaves these quantum dots way too toxic to be safely injected into a bloodstream, scientists looked for a workaround.
To outsource the manufacturing.
Right. To one of the most resilient, adaptable platforms on the planet, they turned to bacteria.
Specifically E. coli. The idea of using microorganisms as green factories for nanoparticle production have gained immense traction lately, precisely because these tiny guys have spent the last 3 billion years figuring out how to survive in heavy metal-rich environments.
The mechanics of how we actually get a microscopic organism to swallow heavy metal poison and then spit out high-tech electronics is just fascinating.
It's all about tricking them, really.
Right. It starts with manipulating their natural biological drives. E. coli are highly programmable. If you want them to do something, you just change their environment. In this case, researchers culture them in a liquid medium heavily supplemented with elevated concentrations of phosphate alongside a highly toxic dose of cadmium chloride.
Phosphate is the bait.
Yeah, because phosphate is an absolute necessity for their survival. They need it to build the sugar phosphate backbones of their DNA, to synthesize the ATP that powers their metabolism, and to construct the phospholipids in their cell membranes.
So when the bacteria sense all this abundant phosphate floating around in their environment, they activate a transport mechanism called the PIT system, the phosphate inorganic transport system.
Okay.
It's a symporter mechanism that goes into absolute overdrive, trying to pull as much of that vital nutrient across the cell membrane as possible.
But the cadmium chloride in the environment throws a massive wrench into the process. Cadmium readily forms a complex with the phosphate, right? It acts as a chemical stowaway.
A deadly stowaway.
As the pit system frantically pulls the phosphate inside to eat, it unwittingly drags the highly toxic cadmium ions right through the cell membrane along with it.
They literally poison themselves by mistake while trying to gorge on nutrients.
It's like eating a really good sandwich, but there's glass in it.
Basically, suddenly the interior of the bacterium is flooded with free, soluble cadmium ions. The glutathione pool is instantly depleted, the ROS levels spike, the zinc finger proteins in their DNA are being displaced. It is an immediate existential cellular crisis.
If the bacterium does nothing, it will rupture and die within hours, right? But bacteria possess incredibly robust stress response networks. To stop the free-floating cadmium from tearing their internal machinery apart, they initiate a chemical lockdown.
And they use the very thing cadmium is attacking, sulfur.
Which is brilliant. The bacteria upregulate specific metabolic pathways to generate sulfur molecules, specifically relying on enzymes like cysteine desulfhydrase. This enzyme breaks down the amino acid cysteine and releases hydrogen sulfide.
That's the compound famous for smelling like rotten eggs.
Yeah, that's the one. So the bacteria intentionally flood their own cytoplasm with this reactive sulfur. Because cadmium has that massive affinity for sulfur, the free cadmium ions immediately bind to the hydrogen sulfide.
This chemical reaction forcefully combines the two, resulting in cadmium sulfide, CDS.
They turn the soluble poison into a solid, insoluble crystal. They lock it down, and that crystal is the quantum dot. It is absolutely brilliant.
It really is.
So the bacteria basically swallow the poison, and to stop it from killing them, they turn it into a glowing pearl. Like an oyster making a pearl out of a painful grain of sand.
That is exactly what it is. And this biological synthesis offers a massive advantage over our human chemical cleanrooms. When we make quantum dots in a lab, we have to use harsh chemical solvents, and the resulting dots are naked.
And knitted dots are dangerous.
Highly dangerous. They require secondary, highly complex processes to add chemical caps to stabilize them. But the bacteria do this organically. As the cadmium sulfide crystal forms inside the cytoplasm, the bacteria naturally coat it with endogenous proteins, peptides, and other biomolecules.
They wrap the toxic rock in a soft, organic, biological blanket.
This biological capping stabilizes the nanostructure immediately. It prevents the dots from aggregating together into giant, useless clumps, and it makes them inherently less toxic than their lab-made counterparts. The bacteria are performing sophisticated green chemistry, entirely autonomously.
It is the ultimate survival mechanism, but as we see in the research, it introduces a glaring logistical nightmare.
The space issue.
Exactly. The bacterium has successfully neutralized the free cadmium into solid quantum dots. It survived the immediate chemical toxicity. But now, it has a physical volume problem.
Because the internal space of an E. coli is incredibly constrained. It is only about 2 micrometers long and maybe half a micrometer wide.
And it is now rapidly filling up with toxic, solid, glowing crystals. It's like sweeping highly radioactive dust into solid piles in the middle of your living room. The dust isn't floating in the air anymore so you can breathe, but you still have a living room full of radioactive rocks.
Right. Eventually, the sheer physical accumulation is going to completely compromise the cell.
So how do they get the garbage out of the house? How does a bacterium empty its internal trash? That has been the central, elusive question in nanobiotechnology.
We could easily observe the bacteria synthesizing the dots, but how they manage the physical burden of the waste was a total mystery.
And this is where we have to shift our focus to a really strange, microscopic anomaly. a biological quirk that microbiologists knew about for decades, but largely dismissed as a useless genetic mistake. The Minicell.
Yes, the Minicell is fascinating because normally the architecture of bacterial cell division is a masterclass in symmetry.
Right.
When a healthy E. coli reaches a certain size and prepares to replicate, it elongates. It duplicates its single circular chromosome, pulls the two copies to opposite ends of the cell, and then finds its exact mathematical center. A protein ring forms, pinches the cell directly in half, and you get two identical daughter cells.
Beautiful, clean division. But the Minicell is the result of that process going completely rogue. It is a non-canonical polar cell division. Instead of dividing down the middle, the division machinery drifts all the way to the very tip of the bacterium, the pole, and just pinches off a tiny spherical bubble.
What makes this structure so unique is what it lacks. Because the division happens at the extreme pole, far away from the duplicated chromosomes, the resulting Minicell contained absolutely no chromosomal DNA.
It is a zombie pod. Without a genetic blueprint, it has no capacity to ever divide, grow, or reproduce. It is just an isolated sphere of bacterial membrane and trapped cytoplasm floating away into the void.
For a long time, the assumption was that this was simply a lethal mutation. We most commonly observed it in strains that had mutations in the MIN division system.
The MIN operon, consisting of 3 vital genes, MINS, MIND, and MINE. The protein oscillation of this MIN system is actually one of my favorite things in microbiology.
It's incredible.
It acts as the bacterium's internal tape measure. The MIND protein binds to the cell membrane at one of the poles and pulls MINC along with it. And MINC is an inhibitor, right? It actively blocks the cell division machinery from forming.
Then MIND comes along and essentially sweeps MINC and MIND off the membrane, forcing them to migrate to the opposite pole. This creates a continuous oscillating wave of inhibitor proteins sweeping back and forth from pole to pole.
Like a pendulum.
Exactly. And the only place in the entire cell where the concentration of the inhibitor is low enough to allow division is the exact dead center.
But if you delete those genes, right, like if you have a MINC deletion or you knock out the entire MINCDE operon, the tape measure shatters, the inhibitor way stops. This cell division machinery, primarily the FTSE protein ring, is no longer restricted to the center. It starts randomly assembling at the poles.
And the morphological result is drastic. The main bacterium fails to divide properly and grows into a long, continuous filament. And at the ends of this filament, it just constantly, rapidly pinches off these tiny DNA-free Minicells.
It is so easy to see why early microbiologists thought this was just a broken machine spitting out broken parts.
Totally.
But science rarely settles for useless. Researchers, specifically looking at the work of Rang and his colleagues, started noticing that these Minicells might actually serve a profound physiological role in stress tolerance.
Rang's team did some brilliant work. They subjected E. coli to a massive stressor, the antibiotic streptomycin. Streptomycin works by binding to the bacterial ribosome, which induces massive errors during protein synthesis. The cell starts churning out misfolded dysfunctional proteins.
The exact same kind of misfolded protein garbage that cadmium causes.
Precisely. And Rang's team made a stunning observation. The bacteria weren't just letting these misfolded proteins float around, they were actively aggregating the damaged proteins and physically shoving them into the poles of the cell right before the Minicells pinched off. The bacteria were using the Minicells to jettison the damaged proteins.
Here's where it gets really interesting. Are we saying the bacteria are basically dropping their tails like a lizard escaping a predator? Except the tail is a tiny DNA-free garbage bag.
That is a perfect way to look at it. It is an incredibly elegant solution for a single-cell organism to invent a localized quarantine zone to maintain the fitness of the mother cell. The mother cell knows that if the garbage builds up, she dies. So she packages the biological waste into a DNA-free pod and ejects it.
Just sacrificing a tiny bit of membrane to survive. It is a remarkable mechanism for maintaining cellular homeostasis under extreme duress. And this brings the threads of our discussion together perfectly.
Right, because we know cadmium causes massive protein misfolding.
We know bacteria crystallize cadmium into glowing quantum dots. And we know Minicells act as garbage bags for damaged cellular components. So the brilliant million-dollar hypothesis is obvious. Are the bacteria intentionally using these Minicell escape pods to physically throw the highly toxic, glowing heavy metal crystals in the trash?
It's a hypothesis that elegantly bridges physics, chemistry, and microbiology. But to prove it, researchers had to design an environment where they could watch the manufacturing process and the waste disposal happen in real time under extreme magnification.
They built a microscopic gladiator arena. They took normal, healthy E. coli, the wild type, heavily researched BW25113 strain. These are bacteria with a fully functioning MIN division system. They do not naturally make minicells.
Right.
Then they compared them side by side with the mutant strains, the ones with the broken tape measure. Specifically, a strain missing the MIN-C gene and one completely missing the MIN-STE operon.
They placed both populations in an M9 glucose minimal liquid medium, which restricts their diet, and supplemented it with a staggering 60 micrograms per milliliter of cadmium chloride. They forced them to manufacture the dots under immense chemical pressure.
And the reaction of the normal wild-type bacteria is a grim look at biological failure. Without the ability to form minicells, they panicked. They were completely overwhelmed by the manufacturing process.
To survive the massive influx of cadmium, the wild-type bacteria initiated a desperate collective defense mechanism. They clumped together tightly and began secreting a massive amount of extracellular polymeric substances.
They built a thick, sticky, sugary biofilm matrix around the colony, basically attempting to trap the cadmium ions outside their cell walls.
They tried to build a bunker out of biological glue, but it was a losing battle. The cadmium penetrated the matrix. Under the microscopes, the researchers observed massive, dense deposits of cadmium forming primarily at the poles of these normal cells. The internal space was maxing out.
The physical toll was catastrophic. The wild-type bacteria exhibited a severe loss of membrane integrity. The cell walls were physically degrading. The cells were swelling, bursting, and dying. The toxic accumulation of the heavy metal crystals was literally tearing the factories apart from the inside out.
But when we shift the focus to the mutant strains, the ones constantly spitting out the tiny zombie pods, the contrast is absolute night and day.
It really is.
The Minicell-producing strains thrived beautifully. They didn't panic. They didn't clump together in a sticky biofilm. They maintained their structural integrity, remaining as healthy, distinct, individual rod-shaped cells.
They were processing the cadmium without suffering the fatal accumulation that destroyed the wild type, because they had a pressure valve.
Exactly.
But the researchers needed definitive proof that the quantum dots were actually inside the minicells, so they turned to the light. Using fluorescence microscopy, they excited the cells with UV light. The main rod-shaped mother cells glowed, confirming the biosynthesis of the quantum dots was happening.
But crucially, scattered all around them, the tiny spherical minicells themselves were glowing bright orange and red under the UV light.
The glowing escape pods are the visual smoking gun. But visual fluorescence isn't enough to definitively prove the chemical composition and spatial location of a 2-nanometer crystal. The researchers had to bring in the heavy analytical machinery, starting with transmission electron microscopy, TEM.
And TEM is fascinating physics on its own.
Oh, absolutely. Instead of using light, which is, limited by its wavelength, TEM shoots a highly focused beam of electrons through a vacuum, directly through an ultra-thin slice of the biological sample.
Right. Electromagnetic lenses focus the beam, and when the electrons hit something dense like a heavy metal, they scatter.
And the unscattered electrons hit a detector, creating this highly magnified shadow image of the internal ultrastructure of the cell.
Looking at the TEM imagery of these mutant strains is a total revelation. Inside the cytoplasm, near the inner membrane, you see hundreds of tiny, distinct, incredibly uniform black clusters. These are the electron-dense cadmium sulfide nanoparticles, perfectly sized between 2 and 6 nanometers.
But the real action is at the poles. The TEM captures the exact moment of ejection. You can see these dark, heavy metal clusters actively congregating at the extreme tip of the cell. The cell membrane is physically distending, forming a bubble, and trapping a dense concentration of the black clusters inside it right before it pinches off.
It is an undeniable visual record of the garbage disposal process.
To confirm the chemical identity of those black clusters, the researchers utilized energy dispersive spectroscopy, or EDS, in tandem with the electron microscope.
Right, because when the high-energy electron beam hits the sample, it knocks inner shell electrons out of the atoms. Outer shell electrons fall down to fill the vacancy, and in doing so, they emit an X-ray.
And the energy of that X-ray is a unique, exact fingerprint for every element on the periodic table.
Precisely. The researchers targeted the electron beam specifically on the dark clusters inside the minicells. The resulting X-ray spectrum showed massive, undeniable peaks for cadmium and sulfur. The glowing dots inside the minicells were definitively cadmium sulfide quantum dots.
They went one step further and quantified the efficiency of the trash system using flame atomic absorption spectrometry. FAS. They literally atomized the separated minicells and mother cells in a high-temperature flame, shined a specific wavelength of light through the plasma, and measured how much light the cadmium atoms absorbed.
Which gave them the exact concentration of the metal.
Right. The FAA's data was the final nail in the hypothesis. By volume, the isolated minicells contained a significantly higher concentration of cadmium than the main rod cells. The bacteria were heavily prioritizing the movement of the toxic material into the disposable pods.
Which raises a massive logistical question about the mechanics inside the cell.
Okay, yes. How does the garbage actually get to the garbage bag? Do the dots just float to the ends? Or is the cell physically carrying these glowing rocks to the exit? Because we are talking about a highly complex, crowded intracellular environment.
It totally defies logic to assume these quantum dots are just randomly floating around and accidentally drifting into the poles just in time to get pinched off.
Right. Active transport is absolutely required.
Exactly. This is a highly regulated biological process. We have to look back at the organic coating the bacteria put on the quantum dots. Because the soluble cadmium causes massive protein misfolding before it gets crystallized, the quantum dot itself is almost certainly nucleated around or enveloped by damaged misfolded proteins.
So it's literally wearing a coat made of biochemical garbage.
It is, and the bacterium has an incredibly robust surveillance system specifically tuned to detect that kind of garbage. The cell relies on chaperone proteins.
Complexes like GRO-EL and GRO-E or DNAC.
Right, these chaperones constantly patrol the cytoplasm. Their primary job is to find misfolded proteins, bind to them, and attempt to refold them into their proper functional shapes.
But a protein wrapped around a solid heavy metal crystal cannot be refolded.
It's unfixable. When the chaperone proteins realize the target is beyond repair, they flag it for disposal. It is highly probable that these chaperones, or similar transport machineries, physically bind to the damaged organic coating of the quantum dot and actively traffic the entire complex along the cytoskeletal framework.
Just dragging it directly to the poles where the minicell is forming.
Treating the dot like a disease.
They march the toxic payload right to the biological airlock. The level of coordination required for a single-celled organism to detect a novel synthetic threat, crystallize it, wrap it, flag it, transport it, and eject it is nothing short of miraculous.
It is a profound demonstration of biological adaptability. But in measuring the success of this trash disposal system, the researchers uncovered an entirely unexpected side effect.
Oh yeah, the kinetics.
A quirk in the kinetics of how these dots grow inside the minicell that completely changes their value to the nanotechnology industry.
This is where the biology of the escape pod directly alters the physics of the quantum dot. We have to look at the timeline of fluorescence, the growth curve of the crystals. Let's start by looking at what happens inside the normal, wild-type bacteria, the ones that couldn't make minicells and eventually burst.
So, in a continuous culture, the wild-type bacteria have no way to stop the synthesis. The timeline tells a story of uncontrolled runaway growth. Remember, with quantum confinement, the color of the emitted light is strictly tied to the physical size of the dot.
Right. Small dots emit higher energy, shorter wavelength light, like blue or green. Larger dots emit lower energy, longer wavelength light, like red.
So for the wild-type bacteria, after about 4 to 6 hours of incubation with the cadmium, the entire colony starts glowing a bright, vivid green under the UV light.
The dots have nucleated. They're small, maybe 2 or 3 nanometers.
But the cadmium and sulfur keep reacting. The internal factory never shuts off. The crystals keep growing layer by layer. By 8 hours, the physical size of the dots has increased, and the fluorescence shifts from green to yellow. By 10 hours, they are larger still, glowing orange. By 14 hours, the internal space is packed with dots glowing deep red.
They are inflating like balloons.
And then they hit a critical physical threshold. The crystal structure grows too large. It surpasses the Bohr exciton radius, which is the physical boundary required to maintain the quantum confinement effect. The electron energy levels are no longer constrained. The band gap normalizes to that of bulk cadmium sulfide.
So the unique physics basically collapse.
Sure.
The quantum dot dies.
The fluorescence completely vanishes, the bright red glow disappears, and the crystals continue to aggregate into massive, dark, biochemically inert black sludge. The wild-type factory fundamentally ruins its own product by over-manufacturing it.
Which is a massive problem if you are trying to use bacteria to manufacture highly specific imaging tools. You need the dots to be uniform, and you need them to stay actively fluorescent. You cannot inject black sludge into a patient. So what did the timeline look like for the mutant strains? How did the dots behave inside the minicells?
The kinetics were radically different. The Minicells-producing strains started the visible glowing process slightly later, around the 8-hour mark. They went through the exact same initial sequence. They nucleated, glowing green. They grew slightly larger, shifting to yellow. They continued to grow, shifting to orange. But then, they did something completely unexpected.
I just stopped.
The growth curve entirely plateaued and never shifted to red, and they absolutely never surpassed the quantum confinement threshold to become black sludge. They stabilized at that exact physical size, maintaining a highly intense, brilliantly bright orange fluorescence all the way up to the 24-hour mark of the experiment.
So what does this all mean? If the dot stops growing, isn't that a failure of the factory? Why are the researchers excited that it got stuck at orange?
If we connect this to the bigger picture, in manufacturing, control is everything. The Minicell naturally provides an isolated microenvironment where the reaction simply halts at a highly desirable, stable size. It acts as a natural pause button, solving one of the hardest problems in nanotechnology, creating uniform, actively fluorescent dots without them overgrowing into useless black sludge.
But the researchers weren't satisfied with just observing the pause button. They wanted to know the chemical mechanism behind it. Why does the crystal stop growing the moment the minicell pinches off from the mother cell?
The most logical assumption points directly to the supply chain. To grow a cadmium sulfide crystal, you need a continuous active supply of bioavailable sulfur.
Right, the hydrogen sulfide generated by the enzyme cysteine desulfhydrase.
Exactly. The researchers hypothesized that when the minicell pinched off, perhaps it simply didn't take any of that critical enzyme with it. The mother cell kept all the machinery. Therefore, inside the minicell, there is no enzyme to break down cysteine, no new sulfur is produced, and the crystal growth stops immediately due to chemical starvation.
It's an elegant, highly logical explanation. It makes perfect sense. The biological garbage bag doesn't need to contain tools to build more garbage.
Except when the researchers actually ran the biochemical assays to detect the presence of the enzyme, it was there.
Wait,
It
really?
was a massive curveball. The assays definitively proved that cysteine desulfhydrase was not only present inside the isolated minicells, but it was structurally intact and fully active. The enzymatic machinery was shipped into the escape pod right alongside the quantum dots.
So the factory floor, the raw materials, and the machinery are all still present, but the assembly line shuts down anyway. Why?
It leaves a profound, partially unsolved chemical mystery. If the enzyme is present, then some other currently unidentified biological factor must be the limiting constraint.
Could it be energy?
It could be an energy deficit, yes.
Yeah.
Perhaps the minicell lacks the ATP required to facilitate the continuous transport of cysteine across its membrane. Without the raw amino acid precursor, the enzyme has nothing to break down.
Or it could be a physical constraint. The sheer membrane tension of the tiny spherical bubble might exert a physical pressure that prevents the crystal lattice from expanding any further.
Whatever the exact missing factor is, the result is undeniably advantageous. The isolation from the mother cell's active metabolism acts as an infallible natural kill switch for the crystallization process.
It's amazing.
It provides us with a methodology to program bacteria to pump out perfectly uniform, actively fluorescent nanoparticles with an automatic shut-off mechanism built directly into the biology.
Which basically catapults us from a fascinating biological quirk directly into the future of human medicine. We've established that bacteria can make uniform, glowing nanoparticles and shove them into biological trash bags. So here's why this microscopic garbage disposal system is about to impact your future healthcare.
It all circles back to the massive problem we discussed at the very beginning of the deep dive, the toxicity. Quantum dots are miraculous for deep tissue imaging and targeted diagnostics, but the naked heavy metal crystal is too aggressive to put into a human body.
Even the biological quantum dots, the ones coded in the bacterial proteins, still have the heavy metal relatively exposed.
They are a massive improvement over synthetic chemical capping, but they still provoke an immune response, and there is still a risk of heavy metal leaching. But the minicell entirely rewrites the safety profile. Think about the physical structure of what the bacterium is ejecting.
It is a bubble pinched off from the cell's own boundary. It is made of the bacterial cell membrane.
It is an intact, fully enclosed, naturally derived biological lipid bilayer. When the bacterium ejects the quantum dot inside the minicell, it is wrapping the highly toxic, dense heavy metal crystal entirely inside a soft biological sphere.
The heavy metal is completely locked away inside a biological vault.
Exactly. This physical barrier prevents any direct contact between the cadmium crystal and the sensitive membranes of human cells, It radically exponentially increases the biocompatibility of the nanoparticle. It takes one of the most toxic elements on the periodic table and turns it into a perfectly safe, highly functional Trojan horse for good.
That is an incredible visualization. Instead of injecting a toxic, jagged crystal into a patient's bloodstream, you are injecting millions of tiny, soft lipid bubbles. The human immune system, the macrophages, the reticuloendothelial system, they just see a familiar lipid structure. The incredibly bright, stable, glowing beacon is completely hidden inside.
The potential applications are staggering. Because the outside of the Minicells is a biological membrane, we have the technology to engineer it. We can functionalize the surface of the Minicell by attaching highly specific monoclonal antibodies or receptor-targeting peptides directly to the lipid bilayer.
You can basically give the lipid bubble a highly specific set of GPS coordinates.
Precisely. You could engineer the surface of the Minicell to specifically seek out and bind only to the unique receptors found on the surface of aggressive metastatic breast cancer cells.
So a patient receives an intravenous injection. Millions of these engineered Minicells circulate through the bloodstream, completely bypassing healthy tissue because the heavy metal is shielded. They follow their molecular GPS, find the microscopic metastatic clusters, and bind directly to the tumors.
And when the surgical team activates the UV or near-infrared light, the tumor tissue illuminates in a brilliant, unfading, highly specific orange glow, driven by the quantum dots safely housed inside the biological bubbles.
It allows for surgical margins that are currently impossible to visualize.
It really opens the door for localized light-activated cancer therapies, where the quantum dot doesn't just illuminate the tissue, but absorbs specific wavelengths of light to generate localized heat, destroying the tumor from the inside without damaging surrounding tissue. The Minicell delivery vehicle is the key that unlocks the clinical application of quantum dot technology.
But there is one major logistical hurdle to this utopian medical future. We noted earlier that Minicells are primarily a mutant phenomenon. Normal, robust, wild-type bacteria don't naturally make them. They use their functional Min system to divide perfectly down the middle. Does this mean we have to rely on fragile, mutated, genetically manipulated bacterial strains to mass produce these vital medical supplies?
It is a vital question for scaling the technology. Genetically modified organisms are notoriously difficult to scale up in industrial bioreactors without them reverting or dying out. But the researchers uncovered a brilliant caveat in the wild-type behavior that solves it.
Really? How does the heavy metal stress change their division?
Well, while it's true that wild-type bacteria normally divide symmetrically, their behavior under extreme stress is highly malleable. The researchers analyzed the protein expression of the wild-type E. coli when exposed to the high concentrations of cadmium. They discovered that the heavy metal toxicity actively downregulates. It suppresses the transcription and translation of specific crucial cell division proteins, most notably FTSZ and FTSQ.
FTSZ is the core component of the division machinery, right? It forms the Z ring that pinches the cell in half.
Exactly. The cadmium itself acts as a chemical disruptor to the normal division machinery. The environmental stress naturally breaks the cell's internal tape measure, even without a genetic mutation in the Min system.
So the presence of the poison naturally forces the normal bacteria to abandon symmetrical division and start desperately pinching off Minicells as a stress response.
It naturally induces the exact mechanism we want.
Yeah.
We don't necessarily have to rely on genetically fragile mutant strains. We have the potential to use robust industrial-scale cultures of wild-type bacteria, allow them to multiply efficiently, and then simply expose them to a specific concentration of metal stress.
The bacteria will naturally transition into nanofactories, synthesizing the quantum dots, packaging them into Minicells, and ejecting perfectly sealed, highly biocompatible medical imaging tools directly into the culture medium for us to harvest.
Which is just incredible.
It's the ultimate biological irony. It's wild to think that our most advanced medical imaging tech, the tools we will use to hunt down cancer cells and map the human brain, might rely entirely on hijacking the panicked, trash disposal routine of the bacteria that give you food poisoning.
This raises an important question, though, and it highlights the incredible untapped potential of utilizing complex biological systems for advanced manufacturing. This mechanism isn't just limited to E. coli. Both gram-negative and gram-positive bacteria across entirely different phyla have been shown to form Minicell-like structures under severe environmental stress. We haven't just found a quirky trick in E. coli. We've unlocked a potentially universal blueprint for natural nanomanufacturing that spans the entire bacterial kingdom.
It has been a staggering journey from top to bottom today. We started with the immense chemical problem of quantum dots, tools that offer unprecedented optoelectronic precision, but are manufactured from heavy metals so toxic they actively melt cellular engines and shred DNA.
The chemical synthesis is fundamentally incompatible with the biological application.
So we turned the problem over to E. coli. The bacteria accidentally ingest the cadmium while hunting for nutrients, and to survive the immediate oxidative crisis, they bind the poison with sulfur, effectively crystallizing the free-floating weapon into a solid, glowing nanoparticle.
They organically cap the nanostructure, stabilizing it and reducing its immediate toxicity.
But left unchecked, the bacteria would gorge themselves until the massive accumulation of glowing, red-shifting sludge tore them apart from the inside out. So they utilized an ancient, deeply conserved stress response. They employ chaperone proteins to actively hunt down the heavy metal crystals, march them to the extreme poles of the cell, and force them into a DNA-free membrane bubble.
The Minicell, a localized quarantine zone that physically ejects the threat from the mother cell.
And in doing so, they inadvertently completely halt the growth of the crystal, locking it into a perfectly uniform, highly fluorescent size and wrapping the entire package in a soft, biological lipid bilayer that effectively hides the toxic payload from the human immune system.
It is a masterclass in adaptation, a biological solution to a highly complex physics and engineering problem, executed perfectly at the nanoscale.
I want to leave you with a final thought to chew on as we wrap up this deep dive. We have just spent a significant amount of time detailing how single-celled bacteria, literally some of the oldest, simplest, and most fundamental life forms on planet Earth, have spontaneously adapted to compartmentalize, wrap up, and discard synthetic, toxic nanoparticles.
It really puts things in
No,
perspective.
it does. These are highly engineered, modern, heavy metal concentrations that these organisms have absolutely never encountered in their billions of years of evolutionary history. They figured out a highly complex, targeted physical defense mechanism to our cutting-edge industrial garbage entirely on the fly. So if a simple, single-celled E. coli bacterium can spontaneously invent a microscopic lipid-bound escape pod to survive the toxic byproducts of human technology, what unknown, highly advanced defense mechanisms are silently triggering, adapting, and evolving inside the trillions of complex cells within your own body right now in response to the invisible modern industrial world you walk through every single day? Think about that the next time you hear the word manufacturing. Keep diving deep.